Hello everyone. Today I will be explaining glycolysis, which is the first step of respiration. Okay, so let's have a quick understanding about this glycolysis process. So first of all, what is glycolysis? It is a student first step in respiration. Okay, means what does it mean, sir? It means suppose you are going to break glucose. So why you are breaking this one? You are breaking this one, student, to get energy. So if you break this glucose, then finally they will convert into pyruvic acid. Means one molecule of glucose breaks into two molecule of what? Pyruvic acid. And finally this pyruvic acid, student, given two options. It is given how many options? Two options. First option, either you can choose for aerobic side, aerobic respiration, and other you can choose for anaerobic respiration. Means anaerobic respiration means here oxygen will be absent. Here oxygen will be absent, and here oxygen will be present. I hope you are understanding till here. I am breaking this glucose into pyruvic acid, and pyruvic acid is given how many options? Either in absence of oxygen or in presence of oxygen. If it is in absence of oxygen, then there are two things possible. Anaerobic respiration, here two type of fermentation I will be talking. Fermentation. This fermentation, student, it will be, that is alcoholic fermentation. What it is? Alcoholic fermentation. Or it will be what? Lactic acid fermentation. What it will be? Lactic acid fermentation. What I am telling you, student? Glucose converted into pyruvic acid, and finally it is given two options, whether go for without oxygen, whether with oxygen. Now if this one going with oxygen, then this pyruvic acid converts into what? Two molecule pyruvic acid convert into two molecule of acetyl coenzyme, right? And this will enter what? This will enter what? Krebs cycle. What it will enter? Krebs cycle. A student, and through this Krebs cycle, finally what will be coming out? CO2 and water will be coming out. Okay, and energy will be also liberated. So this all processes, student, it is what taking place in mitochondria. Where it is taking place? Mitochondria. Is this red color visible? Okay. Next you will see, student, just now I will tell you that from here to here, means this glucose converting into pyruvic acid, this process is called what? Glycolysis. What it is called? Glycolysis. Student, the same glycolysis I will be explaining in today's class. So in the breakdown or in the oxidation of glucose, the first step which is coming, it is what? Glycolysis. So let's discuss about glycolysis. Okay. So let me take this whole part here. Okay. So let's have this one understanding of glycolysis. So it is the first step in respiration. Here, student, what happened? Partial breakdown take place. What take place? Partial oxidation or breakdown, same. Partial oxidation of glucose take place. What take place? Partial oxidation of glucose take place. Where it take place, student? Where is the location? So it is taking place in the cytoplasm. Where it is taking place? In the cytoplasm. Student, this process, it is common. This is the common process. Common process in both aerobic respiration or anaerobic respiration. What type of respiration I am talking, student? Means this glycolysis is common process in both aerobic as well as anaerobic. Sir, how it is possible? Because student, in this process there is neither consumption of oxygen nor liberation of CO2. So here neither it is asking for oxygen nor it is giving what? CO2. Okay, and that's why this is the common in aerobic as well as anaerobic process. Student, let's have another one, another slide, and I will be teaching how many steps are there. So student, total steps if I talk here, so total step how many? 10 steps. How many steps? 10 steps. Out of this 10 steps, student, out of this 10 steps, two type of reactions are there. One is reversible, I will first put irreversible, then I will put reversible. Irreversible and here reversible. So which steps are irreversible? So they are first step, they are third step, and they are the 10th step. They are what? Irreversible, and here this irreversible reaction are remaining. All are what? Reversible reaction. Okay, student. In this one you will see, student. In this glycolysis, ATP. It is used also. It is produced also. So, dear student, you will see used where. So it is used in reaction first and third. Where it is used? Reaction first step, third step. And where ATP produced? So student, it is produced in step 7th step, and 10th step. Where it is produced? 7 it is 7th step, and 10th step it is what? ATP produced and used where? In the first step and the third step. Student, also if I tell, so there are control points. In this reaction in glycolysis. So which step are called control point? So I am mentioning that step first, step third, and step 10. Out of that one, student, NCERT mentioning that this is the most important. This is the most important step, because here one enzyme is there that is called phosphofructokinase. Which enzyme? Phosphofructokinase. This phosphofructokinase, student, it is what type of enzyme? Allosteric enzyme. What type of enzyme? Allosteric enzyme. So let me conclude till here once more. Glycolysis, first step in respiration. Partial oxidation. Here, what type of breakdown? Partial, and location where? Cytoplasm. Common in aerobic as well as anaerobic. Neither you are using nor you are giving. Next, how many total steps? 10 steps. Irreversible, how many? 3. That is first, third, and 10th step, and remaining are reversible. Next, ATP used where? First and third, and produced where? 7th and 10th. Next, you will see control point where? 1, 3, 10. Out of these all, which one is more important? That is third step, which is catalyzed by which enzyme? That is phosphofructokinase. What type of enzyme it is? Allosteric enzyme. What type of enzyme? Allosteric. So let's start a new one then. Let's start the whole process. Let's see this one, how it takes place. So student, I will show you the whole process. It is starting with what? Glucose. Glucose having how many carbon? 6 carbon. So finally, the first step is going here, which is what type of process? Irreversible process. So finally, here 1 ATP is used. 1 ATP is used. And finally, you will see this glucose converted into glucose 6-phosphate. What it is converted into? Glucose 6-phosphate. Now, this is also how many carbon? 6 carbon. Now, this glucose 6-phosphate student, glucose 6-phosphate, it is converted into fructose 6-phosphate. Which compound? Fructose 6-phosphate. Fructose 6-phosphate. This is also how many carbon? 6 carbon. Isomers, na? So isomerase enzyme, hexokinase enzyme, right? Enzymes also is important. You will see this fructose now it is converted into by the use of, this is the first step, this is the second step, and this is the third step. So I told you that in the first step and in the third step ATP is used. ATP is used. So finally now you will see this fructose 6-phosphate. It is becoming what? Now fructose 1,6 bisphosphate. What it is becoming? Fructose 1,6 bisphosphate. Now what happened? Again I will summarize this one, do not worry. So finally this fructose 1,6 phosphate, it is converted into two compound, that is here you will see a student, that is 3-PGAL. What is 3-PGAL? 3-phosphoglyceraldehyde. What is 3-phosphoglyceraldehyde? And here is what? DHAP. What is DHAP, sir? It is dihydroxyacetone phosphate. What it is? Dihydroxyacetone phosphate. Now what happened? This is also 3 carbon. And this is also how many carbon? 3 carbon. Now what will happen? This is getting converted into 3-PGL. These reactions are what? These are reversible. So you will see this is the fourth step. Which step, student? This is the fourth step, and now this is fifth step. Okay? Fifth step, after that, what is happening? This is already how many carbon? 3 carbon. And now this is also how many carbon? So this DHAP converting into PGAL. So now here it will be how many molecules? 2 molecules of 3-PGAL. And finally what you will see? This 3-PGAL, it is getting converted into what, student? 1,3-bis-PGA. It is converting into 1,3-bis-PGA. Means 1,3-bis-phosphoglyceric acid. Phosphoglyceric acid. Here only, students, you will see one compound is there, that is NAD+. What it is? NAD+. And it is converted into NADPH2. It is converted into NADP+, I hope. Sorry, NADH2. My God. Sorry. So finally they will become what? NADH2. NAD plus accepting what? Hydrogen and becoming what? NADH2. Here students, you will see 2 inorganic phosphate given. What is given? 2 inorganic phosphate. Sir, why you are giving this 2 inorganic phosphate? So that this 3-PGAL become how many? 1,3-bis-PGA. Earlier how many phosphate it has? 1. but now how many phosphate it is having? 2. Sir, why here 2 phosphate? But why 2 phosphate you are giving here? Because this molecule, how many? This is also how many molecules? 2 molecules. Okay. So that's why 2 inorganic phosphate given to PGA, and that's why it is converting into 1,3-bis PGA. Now student, what will happen? This is the which step? 6th step. 6th step. Now here one more step you will see. In this step, this is with which step, sir? 7th step. Student, finally this 1,3-bis PGA will become what? 3 PGA. What it will become? 3 PGA. Sir, phosphate bond. Yes, phosphate given to whom? ATP. Phosphate given to whom? ATP. So here how many ATP? 2. So how many ATP produced? 2 ATP produced. How many ATP produced? 2 ATP produced. Sir, which process here? SLP. SLP. What is SLP, sir? Substrate level phosphorylation. Substrate level phosphorylation. Now you will see a student one more time. Let's see. This 3 PGA now getting converted into what? Now getting converted into what? 2 PGA. Converting into 2 PGA. Right now? Achha. Finally, this 2 PGA will be converted into phosphoenol pyruvate. What it is converting into? Phosphoenol pyruvate. Here also, these are also 2 molecules. Here also, there are 2 molecules. Here also, there are 2 molecules. Right now, student? Now, this phosphoenol pyruvate now converting into what? Pyruvic acid. How many molecules? 2. So here also, how many molecules of pyruvic acid? 2 molecules. Here one more time, what is happening? Through SLP, substrate level phosphorylation, 2 ADP converting into 2 ATP. 2 ATP. Understanding? And pyruvate kinase is the enzyme used here. Okay. So let me summarize how many steps here total? 7. Now this is 8th step, this is 9th step, and this is 10th step. So student, you will see here total how many steps I discussed. One more time I will summarize this one. Let's see final. What I discussed here, I told you that glucose is converted into, this is how many carbon? 6 carbon. Converted into glucose 6 phosphate. See, glucose don't have any phosphate now, but with the breakdown of this ATP, phosphate released, and that phosphate is placed on which carbon? 6 number carbon. Which carbon? So that's why it is written glucose 6 phosphate. Means on the 6th carbon of glucose, one phosphate is placed. Same thing. Now it is converting into fructose 6 phosphate. So fructose 6 phosphate, student, it is isomer. So which enzyme? Isomerase enzyme you can write here. Next, fructose 6 phosphate, it is converting into fructose 1,6-bis phosphate. Earlier 1 phosphate, but now here how many phosphate? 2 phosphate. One on carbon number 1, other on carbon number 6. And again you thought that extra phosphate, you took phosphate from where? ATP. ATP having how many phosphate? 3 phosphate. And ADP having how many phosphate? 2 phosphate. So where more 1 phosphate? 1 phosphate given to fructose 6 phosphate. So fructose 6 phosphate becoming fructose 1,6-bis phosphate. Right? Now fructose 1,6-bis phosphate. Here let me write one enzyme also. Very important. It is PFK. Phosphofructokinase. What it is? Phosphofructokinase. And a student, this phosphofructokinase, it is catalyzing this third reaction. Now, fructose 1,6-bis phosphate converting into 3-PGN and DHAP. DHAP means dihydroxyacetone phosphate. This is converting into PGN. And one molecule now becoming how many molecules? 2 molecules. This 2 molecule, student, this 2 molecule 3-PGN, it is converting into two molecule of 1,3 bis PGA. 1,3 bis PGA. In this sixth step only how many? 2 NADH2 produced. How many? 2 NADH2 produced. Now 1,3 bis PGA, it is converting into what? 3 PGA. Means from here one phosphate given. So two molecules now. So two phosphate given and 2 ATP synthesized here. Which process? SLP. Again 1,3 bis PGA converting to 3 PGA. 3 PGA converting to 2 PGA. 2 PGA converting to phosphoenol pyruvate. And this is phosphoenol pyruvate converting into pyruvic acid. That's all. Here you will see a student. After this one, 3 PGA, all are what? 2 molecules. What are they? 2 molecules. So this is all. I must say this is about means a quick idea about what? Glycolysis. Thank you.